Good afternoon, everyone. I am Curtis Busby Earl, and I'd like to welcome you all, whether you're joining us via Zoom or YouTube, to our session this afternoon, the second in our Professor Speaks series. Firstly, just a quick reminder, those of you joining us via Zoom, if you could please remember to keep your microphones muted. And when the time comes and you'd like to ask Professor Weber a question this afternoon, if you could kindly virtually raise your hand and we may then acknowledge you. So this afternoon, it is my pleasure to introduce to you Professor Mona Weber. Personally, whenever I've interacted with Prof. Weber, she's such a calm lady. It's always been such a pleasure listening to her and hearing her speak. And it really is a pleasure, as I say, to introduce her to you this afternoon.
Mangroves.
So Professor Mona Weber is a marine biologist in the Department of Life Sciences here at the University of the West Indies, in the Mona campus. She has approximately 30 years of experience in research and teaching in Jamaica. At present, she is the James S. Moss Solomon Senior Chair in Environmental Management at the UWI, the Director of the Center for Marine Sciences, and the Director of the Discovery Bay Marine Laboratory. Previously, she was also the Academic Coordinator of the Port Royal Marine Laboratory. Mona currently supervises several graduate students in research projects ranging from mangrove and seagrass assessment for the development of appropriate conservation and rehabilitation approaches to mangrove and seagrass carbon sequestration, in particular blue carbon value, and the biodiversity associated with these high valuable ecosystems. Her other research interests include assessments of coastal water quality using plankton and microplastics, and biology and uses of sargassum. So without any further delay, I know I'm far more interested in hearing Prof. Mona speak than hearing myself. So Prof. Mona, the floor is yours. And thank you again for agreeing to join us this afternoon.
Thank you. Thank you, Dr. Busby Earl. And good afternoon, colleagues. Thank you very much for the opportunity to speak on mangroves, microplastics, and sargassum. What is the connection? These areas represent ways that I have been engaging the environment for a long time and engaging students and colleagues to know more and do more in our environment. So I will answer the question right away. What is the connection? These areas show us that our environment is in crisis. Ecosystems like mangroves, coastal lagoons, or oceans are in peril. And we keep using terms like existential threats from climate change, but also from direct anthropogenic human actions that are causing and will cause irreversible change and damage if we don't act now. So I want to talk about how our mangroves are in peril, how our coastlines are in peril through plastics and microplastics, and signs that our oceans are in peril by this sargassum crisis or opportunity that we now face. I will start with mangroves. I think I cut my teeth as a researcher on mangroves. One of my first externally funded projects was the mangrove ecosystem, a biodiversity hotspot. I was very aware. I mean, we teach, we see around us in Jamaica that mangrove losses were dire. Globally, 1 to 2% per year of mangroves are being lost. And these estimates are up to 2010. In some places, it's as much as 8%. And so 20 to 35% mangrove loss up to almost 2010. And these are greater than losses of tropical rainforests and coral reefs. And so knowing and hearing all of this, I couldn't understand why mangroves were not being given the attention, like in many cases, coral reefs. And so I thought if we had a way of bringing the value and the knowledge, the information about the importance of the mangrove ecosystem to the public, then we would help convince and effect the conservation of these environments. One of my favorite quotes that I used to end almost all my presentations on by Baba Dionne is, in the end, we will conserve what we love, and we will love what we understand, and we will understand when we are taught. And so this, I thought, would help to bring over the importance of mangroves by promoting mangrove biodiversity. And the biodiversity project involved documenting, photographing, categorizing all the various species in the mangroves. Through Celia Jackson's work, we were able to identify a new species of sponge. And the idea was to highlight these creatures are not just pretty and colorful, but they had critical value in the whole functioning of the mangrove ecosystem. They're filter feeders. They're the ones that remove particles from the water. They are our natural purification systems. There's food in the mangroves. There are unusual species in the mangroves. There are drugs. Echinacea turbinata, the orange sea squirt, has anti-tumor drugs that it naturally produces. And so the Biodiversity Hotspots project was intended to bring to the fore all the material we often have locked in our theses and publications. Despite this, we were able through that project to support 12 theses and research reports and publications about this biodiversity that we were unearthing. But we were also able through our partners at the Environmental Foundation of Jamaica, really the culmination of this project was a biodiversity center, a special place where we could host the public, bring students, teachers, and families to find out about the biodiversity and the value of our mangrove ecosystems and other ecosystems. And so this continues to be successful. We continue to do what we intended to do, but I became very aware that despite these efforts, responses to mangrove loss and mitigation for damage remained inadequate. And this was along with the realization that we needed to appreciate it. It's more beneficial to conserve than to restore mangroves. And this is a paraphrasing from a Ramsar quote. Because mangrove losses continued. In Jamaica and across the Caribbean, up to, most of these estimates are up to 2010, the Dominican Republic, 3.4% of its mangroves lost. Antigua and Barbuda, St. Vincent, Jamaica. And so we needed to do more than highlight the biodiversity. We are becoming more and more aware globally of the tremendous ecosystem services that mangroves provide, over 200 of them. And so to present them to the public and to bring them home so persons understand the value of mangroves, these services are often grouped into regulating, provisioning, supporting, and cultural services. And especially the regulating services help us to highlight the value of our mangrove ecosystems. These are the biofiltration functions. Mangroves protect coastlines, a very important feature now with the effects of climate change. And another climate-related regulating service is the carbon sequestration, tremendous carbon sequestration function of mangroves. And so globally, we value mangroves economically and ecologically very highly. Has this helped to stem the loss? We will see. In Jamaica, we are taking on the mantle to value our own mangroves. The World Bank PROFOR project, led on campus by Dr. Mandal, saw us being able to give attention to the value of our Jamaican mangroves. In a well put together document, we're able to see how forests like Hunts Bay help to protect lives and property. Flood protection is highlighted, storm protection, and this is in terms of millions of US dollars per hectare. We always accepted that fisheries is an important provisioning function of mangroves, but these documents and these research bring to the fore blue carbon is an important function of mangroves. And so Jamaica's 9,700 and so hectares of mangroves account for 3.7 million tons of carbon, worth 180 million. And this is present value. If we leave our forests alone and keep them in place for 100 years, this value goes up to 17.8 billion US dollars. And many of these assessments didn't include the biodiversity and didn't look at things like ecotourism potential. So we are onto something. We are valuing mangroves differently, and we are valuing mangroves at a time when climate change is upon us. And so part of the mitigation for mangrove loss has to involve assessing factors like blue carbon. I mentioned it before. Patrice Francis is a graduate student who is working on blue carbon, but this is a term that is used for the ability of plants and the sediment, associated mangroves and other coastal vegetation to capture and store CO2. So we're assessing and will ultimately be able to value our mangroves more accurately in terms of their above and below ground biomass and the carbon stored in this biomass. We're developing equations that are specific to our Jamaican mangrove species, so we can quickly and easily value in terms of this metric and perhaps help to convince that it's more valuable to leave the mangrove stand in place than to build a hotel, because we know the actual value of the blue carbon. We are doing a lot of work, and we're proud of our work in mangrove restoration. And this is one of the ways that we're trying to mitigate the loss of mangroves. And Dr. Trench, with his hydrological restoration approaches for degraded mangroves, has gained national, regional, gaining international reputation for not just describing approaches, but conducting actual restoration. And with our partners like NEPA and others, significant areas of the Jamaican mangroves have been restored. But we know that restored mangroves have little stored carbon. Once you destroy a mangrove forest, you lose the carbon that it had stored. And so we're now seeking to combine our blue carbon research with our restoration efforts. What's the rate of carbon sequestration that these restored forests show? Are mangrove forests restored under different conditions, able to pull more carbon than others? And so we're using this knowledge and our research partnerships to see if we can enhance and explain and convince of the value of mangroves. Because we are aware that our restoration efforts, especially in areas like Kingston Harbor and other urbanized coastlines, are often thwarted by the plastics and the contaminants that we release into our environment. So this segues into the second part of my talk. I want to talk about coastlines in peril from plastics and microplastics. And this is not just a Jamaican issue, this is a global problem. It's a Caribbean problem. Caribbean SIDS have been categorized as being among the greatest plastic polluters per capita in the world. We have made the Forbes list. Of the 30 global polluters per capita described by Forbes in 2019, 10 are from the Caribbean. And the top Caribbean plastic polluter per capita releases 1.5 kilograms of plastic per person per day into the environment. And Jamaica didn't make the Forbes list because our population is large, so we didn't make the per capita shortlist. But on events like International Coastal Cleanup Day, Jamaica records the highest number of items of garbage collected for the region, followed somewhat distantly by Puerto Rico and the DOMREP. But we are regarded as the highest polluters based on International Coastal Cleanup Day statistics. And 90% of this material is made from plastic. And Recycle Partners of Jamaica have tried to put things in context for us with recent data on the amount of plastic bottles we use. More than 800 million plastic bottles used by Jamaicans and our visitors. And more than 80% of these end up in the environment. And so we have a plastic problem. And because of that, we have a microplastic problem. We are very familiar, with the impacts of plastics on large marine animals or on fish, on our birds. These are some of the most visible and disturbing images put out by NOAA. The ingestion and suffocation and entanglement of hundreds of marine species, or our own seabirds on the Pedro Bank, we heard recently, are affected by large items of plastic. The plastics also provide transport for invasive species. They've been shown to increase coral disease, and then climate relevant gases, methane and ethylene, are also produced as the plastics degrade. So these have far-reaching environmental issues. We here in Jamaica have focused more on the impacts of the plastics on our coastal plant species. Mangrove seedlings in particular in our restoration areas, we have seen precipitous losses in seedling survival when the plastics surround and smother the plants. We've done experiments in our nursery that show not only are the plastics very damaging in several concentrations, but the items, the plastic bags are the worst. And so we are very aware of how these compromise our restoration efforts. But the plastics also cause damage to existing forests. You've heard a lot, maybe too much, about Refuge Key, but Refuge Key is still our best way of illustrating how plastics can damage forests. And so we Many of you will know the story of Refuge Key having been cleaned of its plastic berm by the efforts of the fishers and the locals in the Port Royal area, and of our attempts to revegetate the key by planting almost 400 seedlings, of which only 33 survived. But the good news is, having removed the plastic berm and having seen the flushing and the water movement with the tides come back to Refuge Key, we have now seen that the forest is coming back. And so October, this month, we were able to see hundreds of natural seedlings and saplings, excuse me, are now growing on Refuge Key. And so we really just needed to reverse the damage that we had done, and the mangrove ecosystem, the forest, would naturally regenerate. But our work in the Kingston Harbor, we have a Kingston Harbor ecosystem assessment management project, and we have been assessing mangroves, especially in Kingston Harbor and Hunts Bay, to see how they can be rehabilitated, how we can mitigate their loss. And it is distressing to see the quantities of plastics in these mangroves. And when you look more closely, you realize that in some areas, the plastics have fragmented, and they look like the sediment on the forest floor. And they will continue to fragment into smaller and smaller pieces, becoming microplastics. And so we have direct evidence that we are in the age of plastics. The abundance of plastics and microplastics globally, microplastics in sediment layers, are indicating that this is a new historical epoch, the Plasticine. In the ocean, we have watched, since 2015, we had only 3% of the plastic in the ocean by weight being microplastics. By 2018, it was 50%. And now we think 99% of the plastics in the ocean by weight are microplastics. And we are beginning to study microplastics. The globe, there are more publications on microplastics than you would imagine. And we realize that the types, the categories of microplastics we see in our environment is related to the type of waste we generate and how we manage our waste. And the takeaway is, the issue is, plastics and microplastics are toxic. The chemical constituents in plastic, what makes up the plastic, is toxic. And plastics absorb, or they take up, chemicals from the environment. And so experiments have shown that new plastic, virgin plastic, does not transfer as much toxins to fish as plastics that have been in the environment and have sequestered all these other toxic material. And the problems have been documented. The chemicals are carcinogens, they are endocrine disruptors, they cause metabolic disorders. And if you burn the plastic, you release even worse chemicals that cause harm and death. Plastics and microplastics in the sea, and in particular microplastics, these small fragments are fed on by our marine animals. They filter feeders like our plankton. and the deposit feeders in the sediment mistake these small fragments for food. And so there are issues now with chemical accumulation. The chemical issues involve the accumulation and bioaccumulation of the food chain, from the plankton to fish and eventually to humans. The microplastics also cause physical issues in these animals, digestive tract blockages and internal damage. And so we run the risk of compromising our food chain, both physically and chemically. Microplastics are found in marine species all over the world. Oysters, crabs, shrimp, sea cucumbers are all species from which quite significant amounts of microplastics have been extracted. And the effects include reduced food intake, reduced processing of food, and eventually smaller body sizes in these organisms. And this has the potential, of course, to impact our fisheries. Our fish yield is affected by smaller body size of these species. And so right here in Jamaica, in Kingston Harbor, we sought to do our own research, starting with finding out how much microplastic we have in our waters. And our Kingston Harbor, being our most polluted bay, was the place to start. The study was really the idea, the brainchild of Deanna Rose, and we worked together across the Department of Life Sciences, across ISENS, across chemistry. We did the microscope work in life sciences, identified and counted and measured the microplastic fragments. ISENS helped with the extraction and other aspects of the research, and chemistry helped us with the analysis of the polymers, the dominant type of polymers in our plastic samples. And so we learned that fragments, the blue area of the pie, indicate that fragments are the dominant type of microplastics in our waters. And this is expected because these are breaking up from plastic bottles and plastic bags. We don't have as much fiber, and we have far less flakes, or we have far less foam or microbead fragments in our waters. We were able also to identify that polyethylene and polypropylene dominated the polymers, and these are typically the PET bottles. Small sizes also is important because it means the plastic is fragmenting. And as it continues to fragment, microplastics will become nanoplastics, where they can be even more damaging in our environment and in us. So I want through this to highlight the importance of research partnerships, and it's critical that we do research. While we recognize at the university that we cannot directly stop the plastics from getting into the Kingston Harbor, we believe we can do our part by doing good research. Research can inform policy. It can help us educate the population, as well as help to show the effectiveness of the interventions. We recognize that we need good research partnerships with our colleagues, international colleagues, the IAEA and ISENS and NEPA. We're working together to do studies on our own bioaccumulation in our food chains and food webs. We're working to elucidate the connectivity between the sediments and the water column and the animals. And we are highlighting the fact that we need better equipment and better training to deal with these issues in our own university. So we know we need to turn off the plastic tap. And we are hoping to convince our regulators and our consumers and our merchants to work to have this done, because we have to clean up the environment. We have to remove the plastics before they become microplastics. And I am encouraged. I decided to introduce this partnership, really being driven by the Grace Kennedy Foundation and Caroline Mahfood partnership to actually clean the Kingston Harbor. The Ocean Cleanup and International Agency, along with the Grace Kennedy Foundation and local organizations like Clean Harbors and Recycle Partners of Jamaica, are working together with other partners to actually show how we can remove the plastics or help to reduce the plastics that are entering our harbor. And so we are hopeful. We need to move from science to action, as was said in a recent Lena editorial. And so we look forward to seeing how this will help in the mitigation of the plastics in our environment, because it brings to the fore how our inputs, what we release into the environment, and how these substances can cause serious problems in the long run. This brings me into the section on Sargassum seaweed. And we are still understanding how Sargassum seaweed has come to be in such large quantities in our oceans and eventually coastlines. But the evidence, the issue is stemming from our inputs into the environment. So we started seeing sargassum on our coastlines in the Caribbean in 2011. 2015, 2018, 2021 were particularly bad years. This year, of course, particularly bad years. We still don't quite understand why the sargassum is coming. We know sargassum is normally from the Sargasso Sea. And we know there is now a new Sargasso Sea that has formed just outside the Caribbean, a little south of that. And researchers like Brian Lapointe and his colleagues have recently said the science is still not definitive. After 10 years, we're still not absolutely sure. But the research suggests that, yes, climate change, climate triggers, but also nutrients from Amazon rainforest destruction and land use changes, and even Sahara dust. Those factors have come together to cause the Sargassum problem. And so it's hypothesized that during very heavy storm activity in 2010, climate-induced storm activity in 2010 caused sargassum to move from its normal location in the Sargasso Sea and to be deposited near the equator. North Equatorial region, recirculating region, received enough sargassum to have a nucleus. And this was driven by climate-induced changes. But the algal mass was fertilized and maintained by nutrient inputs. A lot of the Amazon rainforests were being cut down, and farming was intensifying. Intense rainfall caused by climate change over the Amazon basin acted with that to deposit huge quantities of nutrients into the sea. Even in the Caribbean, our own activities are depositing huge quantities of nutrients. Lapointe and his colleagues indicate that nitrates in the Caribbean are now 35% higher than they ever were. The ratios of nitrates to phosphates are 110% greater. So our actions are also exacerbating and causing these problems. This is how we have over 20 million metric tons of Sargassum biomass moving through the Caribbean in 2018. And so what is the solution? The point says we need to change our policies. We need to just treat our sewage and stop cutting down trees. And we wish it was as easy as saying it. But until then, the realization that sargassum is valuable algal biomass has taken off across the Caribbean and Gulf of Mexico, and the use of sargassum has exploded. The CARDI in 2015 was one of the earliest organizations to put out a policy brief indicating that sargassum seaweed had value in crop and livestock production and could drive agribusinesses. In 2020, our colleagues in Barbados have put out a comprehensive sargassum uses guide, and they have highlighted over 14 different uses to which you can put sargassum. Agriculture, biofuel, biogas generation, environmental restoration, production of paper and bioplastics are some of the important and noteworthy uses that sargassum can be put to. And so in Jamaica, like the rest of the Caribbean, we have been experiencing these deposits of sargassum. In 2014-15, it was really a problem on the North Coast. In 2018, sargassum was everywhere, and it sparked interest from several sectors to see what we could do with this sargassum. In this year, we have noticed huge deposits of sargassum in places like Helsha, and these are similar to the quantities we had in 2018. In fact, in May of this year, we actually saw 6% more sargassum biomass than in the equivalent month in 2018. And so we recognize that the material can result in grave economic losses across the Caribbean. This realization is very clear. Tourism and fisheries, and just the effort to clean the beaches and to contain the material has huge places huge economic burden on our countries. Recreational activities are affected. There are fish kills. Sargassum, when it rots, releases toxic gases like hydrogen sulfide. Smells like rotten eggs. Sargassum buildup on the shoreline causes beach erosion, and sargassum transports plastics and affects or helps to cause biodiversity loss. And so we recognize we had to deal with this material. And like us, the reactions across the globe have been varied and contrasting. Most people want to figure out how to get rid of it. A lot of people are now trying to figure out how to use it. And in 2018, NEPA approached our faculty and we partnered to explore how to create value-added products from sargassum. Our mandate was to explore the potential and commercial uses of this sargassum. And so we went about studying sargassum. We collected sargassum from all around Jamaica, as many shores as it was shown to engulf. We studied the species. Being biologists, we recognize that there are distinct species in the mass, 2 species, and 1 species has 2 types. And these are peculiar to the Sargassum masses that are moving through the Caribbean. We were able to distinguish these by their gross features. But even more importantly, we were also able to recognize that they have distinct chemical profiles. And here we partnered again. The assessment of the chemical profile of the Sargassum was done by our partners in MIAS, Dr. Howard Reed, and we were able to see that with respect to nutrients and alginates and phycoidans, valuable compounds, the Sargassum species and types had different quantities. And we were able to identify which types, which species had the more value-added compounds. And so this research led the MIAS and the University of York, our current partners in this effort, to look at different ways of valorizing, of using Sargassum. So there needed to be bulk uses. There's so much of the material, we needed large volumes of it to be used. And the idea was using it in biomethane production. The idea under this category was using it as fertilizer and compost, as mulch. And this would enhance the growth of plants with relatively little use of synthetic fertilizers. The compounds like alginates and these cell wall polysaccharides that are very valuable in the food industry. These can also help in the production of plastics, bioplastics, good plastics. And thirdly, they showed that in low volume, we could extract quite high value substances, bioactive compounds from Sargassum. So we set about with partners, we have a running project, SARTRAC project, that is aiming to show how we in the Jamaican context can use Sargassum. Partnering with the Scientific Research Council, we are currently doing biomethane potential assessments. They have completed the benchtop experiments, and we've seen good yield from Sargassum mixed with pig slurry. Sargassum on its own does not generate a good yield of biogas. And now we're going to scale up and perhaps have pilot phase use of the Sargassum to generate biogas. Closer to home, in life sciences and botany, we look at Sargassum compost on crops like corn. And there was good growth of the corn, but the marketable yield was not significant. We have tried looking at liquid extract of the Sargassum. And here we worked with UTech partners, students, who compared the yield from the Sargassum as a liquid fertilizer with synthetic fertilizers and of course a control. And the corn and the tomato showed significant improvement, significant growth with Sargassum as liquid fertilizer. So we expanded this to our mangrove work. Could Sargassum help in our mangrove restoration activities? Not under wet conditions. The use of Sargassum compost in the wet nursery led to the production of hydrogen sulfide gas under these wet conditions. And very quickly, the plants in 100% and 50% compost succumbed after 3 weeks. But in the dry nursery and under dry conditions, like replanting along Kingston Harbor's shores, there is potential for use of Sargassum compost. We got good growth in 75% Sargassum compost treatments, and the plants grew well and showed lateral branching. But in 100% Sargassum, we saw negative effects, deleterious effects, including loss of succulence, curling, and discoloration of the leaves. And so we are cautious. We are doing tests to see if chemicals in the Sargassum would have a bad effect on the plants. So those research data will be published. We are also looking at, or have looked at through, again, partnership with the Natural Products Institute, how the smaller quantities of Sargassum and the targeted species, specific species and types, have now been tested for their organic, their bioactive compounds. And these were tested against prostate and breast cancer cells that were cultured by the NPI in their lab. And some potency was observed. These experiments need to be refined. And the particular species, the Sargassum fluitans, proved to be more effective in this regard. And so we know there is potential, but through this research, we also identified challenges. There are harvesting challenges, and our colleagues across the Eastern Caribbean will tell you the volume of material to deal with is very difficult. So there needs to be affordable technologies to harvest Sargassum before it gets to the beach. It's best collected at sea, where before it beaches, because removing Sargassum from the sandy shore often damages the shore. We need to predict where and when the Sargassum will come so that we can more effectively and focus our efforts at efficient collection. And so through the SARTRAC project, Mona Geoinformatics is working on identifying beaches where we are most likely to find Sargassum. Processing for use is another challenge. We recognize that Sargassum picks up heavy metals and toxins from the environment. It has naturally high arsenic levels. And so, and this is the inorganic arsenic that is quite damaging to humans. And so we have to find ways of dealing with those processing issues. The episodic occurrence of the Sargassum, both in terms of quantities sometimes and in terms of quality, can be a challenge. So we are exploring the feedstock, literally, to see how it changes throughout the season and how we can store it and still use it. And so the collection challenges are expensive and really need to be worked out across the region. The feedstock and storage challenges we are attacking through our experimentation. So our collections in 2020, we are now producing a publication. It's been submitted, looking at how the biochemical composition, how the valuable compounds in the Sargassum are changing depending on where they were collected around Jamaica and how they were handled in terms of storage. And so these will inform our valorization approaches as we seek to use this tremendous algal biomass. I read recently that India is culturing seaweed to do some of the very things that we are using the naturally occurring Sargassum to do. And one company, C Combinator, they are a group a public benefit corporation that has developed around looking at how sargassum can be used. And so they are producing a range of products, biostimulants for plants, emulsifiers, leather, rubber, bioplastics. And they would like to expand this industry across the Caribbean. They would like to set up sargassum processing plants on as many islands who are willing. They are particularly interested in marketing the biostimulant because it uses large quantities of the sargassum. And so they will set up factories to produce biostimulant on any island willing to partner in this venture. But the group also believes that along with valorization and using sargassum, the algal biomass actually also produces some of the solutions to the threats and the issues we face in our oceans. If we use sargassum to make biodegradable plastics, we reduce the quantities of microplastics in our environment. If we use sargassum in our soil restoring activities and as plant stimulants, we get better plant growth that removes carbon dioxide. And we use less fertilizer. So we add less nutrients to our oceans. And if there's biomass left over, that can be pumped to the deep ocean and will actually remove carbon from our surface waters. And this is an experiment that has been done by a student from MIT, where he actually demonstrated how much carbon we could remove by pumping sargassum from the surface. And so connecting mangroves, microplastics, and sargassum shows us, yes, that our seas, our oceans are in peril. And it's not just climate change, something that's kind of removed from us. It's our actions. And so we can use what is before us. We can use our knowledge and our research to take action. And the time is now, because these issues that can be turned into solution are before us now. The climate crisis and the environmental crisis that we have is before us now. And so we need to take action before it's too late. I thank you, and I thank our partners for funding and for technology sharing and for helping us to learn a lot. And we hope to do a lot to help our environment. Thank you very much for listening.
Oh, thank you very much, Prof. Mona. I must say, I found it a very, very, as usual, informative presentation. Furthermore, so many of those images you shared were astonishingly disturbing, with the plastics and the sargassum collected on Jamaican's shores. I know you mentioned about the other islands in the Eastern Caribbean. I'm sure that if you were to see some of those images, they would be equally disturbing. I have lots of questions just on my own, but I'm going to start by raising some questions that members of our audience have asked. And in, of course, some cases, I'm hoping that the question has enough that you'll be able to provide an answer to. So firstly, does the return of some of the marine animals indicate that our pollution level here is decreasing in Jamaica?
Sorry, could you start that again? If or what?
Sure. Does the return of some marine animals indicate that our pollution level is decreasing?
Return of some marine animals. I'm not sure of the
origin. Yeah.
Evidence for that.
Mm.
But yes, I mean, it's if we stop what we are doing to the environment. If we reduce our inputs, I guess refuge key might be the context for that. If we fix the problem that we have caused, the systems will naturally regenerate and return. I mean, there was some anecdotal evidence to that after the pandemic, at the start of the pandemic, when humans were removed from the environment and locked in their houses, and the environment water quality began to improve, began to return in a very short time. And it's a good question, because it gives you encouragement and it gives us hope that if we can deal with the problem, the environment will right itself very quickly.
Okay, and related to that question, I was wondering, here in Jamaica, as you know, we've had a ban on certain plastics over the last couple of years at least. Do you see any impact that has had by chance?
That's a good question. We don't have the evidence where we often don't collect and categorize garbage, but we realize we need to do some of that now. But we have seen the reduction in plastic bags in the mangroves. You're seeing the plastic bottles have taken over, but there are far fewer scandal bags reaching the mangrove areas.
Okay, that's at least positive, I guess. Again, related, and something that I've thought, and I'm going to get back to some of the other questions. I think some of it is also cultural. I still see a number of people traveling in vehicles, tossing, what we call juice bags and so on, bag juice and bottles and so on. And so there's still this culture, I think, of littering. Is there anything that you think we could do to address it, to sort of engender change in our culture here? Maybe promoting or displaying or sharing more of the information you've seen. I mean, those images really say a lot about what's happening.
And some of the work we do in terms of the Biodiversity Center and the tours that are done out of the Royal Marine Lab and so on, we get an opportunity. I think there is need for a lot more, but we get the opportunity to show everybody, to show the children, the teachers, persons who we think will be able to help to change the culture. We need to make this sort of exposure. The International Coastal Cleanup Days have been very influential at showing persons the gravity of the situation. And so activities like that, unfortunately, have ceased because of the pandemic. But the exposure of young people and of children and persons who also are the consumers.
Yes.
And the knowledge of how their attitudes and the power that a consumer has. Those things we are working on using those strategies to help with the problem. Okay.
I see I have a question from a member of our Zoom audience. So Professor Kaur, you have a question or a comment?
Yes, thank you, Chair. Thank you for the talk, Professor Weber. I actually was curious about the two questions related to the microplastics in the ocean. The first one, I wondered how they estimated that, those proportions that you gave. And the second one, I wondered what techniques, if any, are there for trying to clean it up, if any, or what efforts might be ongoing to make an attempt to whether recover it, remove it, mitigate the impact. Thanks.
The recovery, I'll start with the second part of the question. The recovery of microplastics. I have not seen any research, any techniques that are being developed to recover microplastics. if you think about the material, it's the size of the plankton, or it's the size of the sediment. Once it is in the environment, and it's everywhere, it's in the deep sea, it is gone. I do not know of any method, and I have not seen anything in the literature indicating how microplastics and nanoplastics can be recovered. And this is why I was a little bit skeptical about cleaning. When you clean up the plastics, they're just going to come back again the next day. But what I've seen in the mangroves, where if you don't remove the plastics when they're big enough to be removed, they're going to fragment, and then you won't be able to remove them. And so prevention is the only solution I know of. With respect to the estimates, now these are global estimates based on not necessarily direct research, but based on literature values of quantities in particular areas. And these are multiplied up, or models are used to scale these up to come up with the quantities. And I think whereas the science might not be perfect, as long as we use these consistently over the periods, then I think the data, the results are comparable.
Right.
So I'm sure as we improve, we will have better ways of doing things, but the consistency of our methods and good methods would support maybe not the absolute values, but the relative values.
Right, I understand. Thank you very much. I wondered whether research into bacteria, things that might be able to metabolize the microplastics might be an interesting way forward.
Absolutely, and there is some of that research. In fact, they can use bacteria now to degrade plastic and turn it into bioplastic.
Right. Thank you.
Okay, thanks a lot, Prof. Kaur. Prof. Weber, there are two other related questions from our social media platforms. One is, are mangrove restoration efforts keeping up with the loss of mangroves in Jamaica? And what I believe is also related is, are mangroves actually protected? Is it a protected species? And if not, why not? And how is it possible for that to be changed?
Wow. Okay, thank you. Thank you for those interesting questions. Mangroves are protected in the sense that you need a wetland modification permit to make any alterations to wetlands. And so usually this captures the large projects that involve tourism infrastructure or road works, any construction that is going to affect mangrove area in Jamaica needs to go through NEPA. And so very often they prescribe that whatever, there's no net loss. There's a no net loss policy. And so if it is impossible to not through the road cause some loss of mangrove area, then an equivalent area has to be restored. Those are some of the most obvious encroachments on mangroves. Sometimes there is, for example, a road that is built and it damages very little mangrove, the footprint, but it interferes with the flow of water from the sea to the mangrove. And so that road can cause acres of mangrove loss eventually, but that sort of thing isn't captured because the footprint isn't affecting the forest. So there should be a way, and I know performance bonds and so on are a touchy issue right now, but there should be a way to have funds held in reserve to mitigate, put in culverts if the culverts were inadequate,
Mhm.
or to do remedial action to stop the mangrove loss by or construction. And the first question, the first part of your question, I've forgotten.
Oh.
I'm sorry.
That's all right. Restoration efforts, are they up
Are they keeping up?
with the loss?
They are, and I think some of the recent research suggests that we are increasing in mangrove acreage, and we think some of that is from the restoration efforts. But as I said in the talk, you get back the tree cover, you get back the seedlings, but you don't get back the carbon.
Right.
The stored carbon below the forest. And so that needs to be factored in when we're talking about no net loss.
Yes.
Not just no net loss of trees, of acreage. It should be no net loss of the stored carbon.
It's still a far better approach to conserve than to restore.
Absolutely.
Okay. I had seen Dr. Singh Wilmot's hand raised. You still have a question, Dr. Singh Wilmot? Mohammadine still with us? Okay, I'll give us some time. Another question that was raised, are phthalates used in neoplastics?
Are what? Sorry?
Phthalates. Yeah.
I'm not sure at all. Like I keep seeing that phthalates and BPA are still an issue. I'm not sure if they're still being used as additives in plastics. The word has gone out that you have to be BPA free. And those sorts of chemicals that are endocrine disruptors, and those are the real nasties that are added to plastics to stabilize them. And they, as far as I'm aware, the word is out that these things should be avoided as much as possible. I don't know if that is so. Okay.
Another question that I have, recycling. I know that that's internationally an approach to keeping or having some sort of control over our plastics. What services are available locally, if you know of any? And again, that may be something that we could, an approach to changing our culture to recycle instead of burning or simply disposing on our hillsides and our beaches and the like. Do you know of any recycling services that are available?
Yes. And I would like to add that I don't know how many of us are aware, but a part of the NEPA plastic ban, the national plastic ban really, involved a scheme to deal with bottles. And bottles are the most recyclable part of our plastic waste. And so Recycled Partners of Jamaica and others are to be funded, and I think are being funded to ramp up the collection, to facilitate the recycling, effective recycling, or at least collection and removal from the waste stream of plastic bottles. Now, I have seen in my own community, communication goes out and information is shared that we're collecting on a Monday, and we're given special bags and so on. That didn't exist before. So based on my own experience, I think there is some increase in the collection of these plastic bottles. It needs to go out. It needs to be in all communities and all levels. We need the action spread.
Okay, and what about so there's been quite a bit mentioned about Jamaica, understandably, but as you've also said, it affects all of us here in the Caribbean. Do you know of any coordinated efforts that exist across our region here in the Caribbean to deal with, would it be the collection or more of the research that you're conducting to deal with sargassum in particular, and with the microplastics?
Yes, or UNEP. UNEP has been doing a a lot of work in trying to coordinate activities, both with respect to plastic pollution and with respect to sargassum, and making sure that the benefits, let's say, or we spread the ability of all countries to be able to deal with those issues, and not just the countries that can afford it. So the technologies and the strategies that are being developed, UNEP is doing a lot to even the playing field across the Caribbean in that regard, because you know the Caribbean is quite a diverse space.
Yes.
And it's very important to try and ensure that all countries are able to benefit from the knowledge and the technologies that we develop.
Okay, that's very informative. That sounds like it's, again, some stuff is very productive. And another question now, this again is personal in the sense that I like, as many of us here in Jamaica, to go to Hellshire or to Fort Clarence or to many of the other beaches, and there's nothing as nice as a fried fish or a steamed fish and so on. But with the fish ingesting the microplastics and so on, I was just wondering about the, let's call it the density. Is it that all of the fish, have there been any studies to get an idea of, is it 90%, is it 50% that we find some alarming level of microplastic ingestion, or are we not there as yet and we're at the stage where we can actually prevent the worsening of that kind of ingestion?
Good question. We are just beginning to do some studies with partners. ISENS and NEPA approached us in the CMS about coordinating efforts to collect species. We're using oysters right now because they're a lot easier to collect, to show the levels of plastic contamination in the mangroves and in the Kingston Harbor in particular. And the interesting thing about the oysters, you see, you tend to clean the fish, you remove the gut. We eat the oyster, we only do not eat the shell.
Right.
You know, everything else is consumed.
Everything else is edible.
And so the threat of actually taking in microplastics from that source is high. And so we're working on collecting and assessing oysters across the whole mangrove area. In terms of finfish, in terms of it sounds bad, but the heavily polluted bays like Kingston Harbor, there isn't a lot of fish. They're juveniles, and the fishers collect bait to go offshore. But a lot of our fishing now is being done further offshore. And perhaps that will give us a chance to catch up with the techniques and the technology to assess finfish more readily, not just for fragments of plastic, but the chemical contamination that will result from bioaccumulation. So we because our coastal waters have become so overfished, there is relatively little fish close enough to the plastic sources, but there are other types of edible resources that we need to know about, we need to safeguard.
Oh, thank you very much. I see another question that is coming from and I won't keep you much longer, Prof. Mona, but from our social media audience, in regards to the Kingston Harbor Cleanup Project, with partnerships you mentioned, and given the lack of garbage sorting currently, are there plans to implement sorting mechanisms that can be used to potentially collect recyclable material and reduce the quantity going into landfills?
Yes, indeed. As part of that project, I'm assuming. Yes.
Yes.
Thank you for the question. And yes, the project will have a landing site associated with it, where the plastics will be handled. And so, because Recycle Partners of Jamaica are involved and others, the intent is to recycle the collected plastic not just put it in Riverton.
Okay. And microorganisms can sometimes adapt to changes in their environment, like using certain contaminants as food. Is there evidence that microorganisms could be adapting to make use of some of these nanoplastics as a part of their nutrient source?
That's an excellent question. There probably is natural use of the material by some microbes. And as you have certain resources become more abundant in your environment, it follows the microbes will also multiply. And the fact that especially the Asians are doing some good research on bacteria and fungi and microbes that can degrade plastics and convert it to usable material or simply convert it to innocuous material. The fact that that research exists means that the possibility is there. And I have no doubt that I live in hope that natural microbes will help us solve those kinds of problems.
Okay, and I think I'll make this the final question. Is the recycling of plastics cost effective as far as you know?
I'm sure that's an excellent question for somebody like Damien King. I am not sure. We have heard a lot about the market. We were able to compress and bale or plastics and sell them, or recycled material was being sold. That really varies according to the market. There's been a lot of talk about controlled incineration, and in some parts of Europe, this is one of the ways that they're using the material. I mean, it's from petroleum, so why not use it for fuel? But you have to be very careful to have the scrubbers and the things to clean the emissions from these incinerators. So potentially, and at some point, there was good value for collecting and selling plastics. I'm not sure what the landscape is like now, but I think we're smart people. I mean, we're going to the moon and other places. I think we can find ways to develop the technologies and the types of incinerators and so on to effectively recycle these plastics and put them back into fuel.
Sorry, Prof, I seem to have lost connection momentarily there.
Okay, I wondered if it was me.
I wondered myself who it was.
Probably means your time is up.
Yes. So, well, now, thankfully, as it was the last question that we'll take this afternoon. But once again, thank you so much for a very interesting and very informative presentation and discussion, and for answering all of the questions. I'd like to say a special thank you to all our colleagues who joined us today on Zoom, and the members, of course, of our audience who join us on the other social media platforms. And I'd like to remind you all to join us for the next Professor Speaks lecture, which should be in November, all things if they go according to plan, and to continue to follow us at the Faculty of Science and Technology on our social media platforms such as Facebook, Instagram, and YouTube. And as well, I'm hoping, I haven't seen that there were members of our audience from our sister campuses and from other islands in our Caribbean region. I'm hoping that we had at least a few. And so thank you again to everyone for deciding to spend some time with us this afternoon. And I look forward to seeing you all again in November. Please enjoy the rest of the afternoon. Stay safe and see you again soon. Thanks again, Prof. Mona.
You're very welcome.